Good evening, and welcome back to Science Me to Sleep, the channel where we take the vibrant, chaotic universe and distill it into a deeply soporific list of facts. If you are currently struggling with the burden of consciousness, you have come to the right place. Tonight we are taking a deep dive into botany, the science of plants. Quiet and stationary organisms, they have mastered the art of doing very little, very slowly. Our placid journey will take us to the highs and lows of the plant world as we focus specifically on growth. I sincerely hope that listening to the minutiae of how grass grows will be just as boring as watching grass grow. Let's begin, shall we? Part 1. The Engines of Growth, Meristems. Sorry, before we get to Part 1, let me note for all the plant biologists listening that yes, I am aware that this is all a grave oversimplification, as gloriously sleep-inducing as it may be to cover an entire college textbook worth of information on this topic. Even I have my limits. Right, on to Part 1. A plant does not grow in the way an animal does, by getting proportionally larger all over. A plant's growth is a more modular, repetitive, and localized affair. It is restricted to very small, specific regions of undifferentiated cells. A cell is called undifferentiated when it lacks the specialized structures or functions that determine its set role within the organism, a bit like young people who have yet to decide what they want to be when they grow up. Anyway, these regions of undifferentiated cells are called meristems. Meristematic cells are the plant's equivalent of stem cells. Think of a meristem as a permanent zone of cell generation, a small self-perpetuating front of new cells, like a biological 3D printer, operating at a very, very slow speed. We will primarily concern ourselves with two types of meristems based on their location and function. First, the apical meristems, apical meaning at the apex or tip. These are responsible for primary growth, which is growth in length, elongation. As you may know, plants grow both upwards and downwards, so they have two flavors of apical meristem. One is the shoot apical meristem. This is a tiny protected dome of cells at the very tip of the stem, hidden within the terminal bud. It is responsible for creating all the upward bound parts of the plant: new stem, new leaves, and new buds. However, the hardworking cells in this part of the plant aren't all mingling together in an open plan office. The shoot apical meristem is comprised of several distinct departments. Its highly organized, layered structure is typically described using the tunica corpus model. The tunica consists of the outermost cells arranged in two layers. L1 is the very outermost layer. Its cells divide only anticlinally, that is, perpendicular to the surface, like how you would slice a bundt cake. This division pattern allows the surface layer to expand without breaking. The L1 layer is destined to become the epidermis, the protective outer skin of the plant, and is therefore often referred to as the epidermal layer. L2 lies just beneath the L1 layer. It also divides primarily anticlinally. The L2 layer gives rise to some of the internal tissues of the plant, and is also referred to as the subepidermal layer. Beneath these layers is the corpus, which includes the L3 layer. This is a bulky central mass of cells. Unlike in the tunica, corpus cells divide in all planes, anticlinal and periclinal, or parallel to the surface. This multidirectional division is what adds bulk and volume, building the internal core of the plant. The L3 layer is responsible for creating the central parts of the stem, including the all-important vascular tissues, the xylem and phloem, as well as the pith at the very center. This structured system allows the meristem to precisely build the complex three-dimensional structure of the plant from a single tiny point. A moment ago I mentioned xylem and phloem, two terms you may yet to have had the pleasure of becoming acquainted. They comprise the transport tissue, aka plant plumbing. The xylem primarily carries water, but also certain nutrients, upwards from the roots to the rest of the plant. while phloem's job is to carry food from the leaves, where it is produced through photosynthesis, down toward the roots, which work up quite an appetite, toiling away in the dark. Speaking of which, we come to the root apical meristem, located, unsurprisingly, at the tip of the roots. It too is a dome of dividing cells, but this one is protected by a small disposable layer of cells called the root cap. The root cap acts as a sacrificial helmet, sloughing off cells as the root relentlessly pushes through the abrasive soil particles. The root apical meristem is also organized into various departments that give rise to various parts of the root. However, they seemed even more complicated than in the shoot apical meristem, as well as harder to pronounce, so we'll just skip that part. Behind the root cap, we find three distinct and reassuringly predictable zones. First is the zone of cell division. This zone includes the meristem itself, and, as you may have guessed, is where all of the cell division takes place. Second, the zone of elongation, where the newly created cells stop dividing and begin to stretch. This stretching, which occurs via simple hydraulic inflation, is what actually pushes the root tip forward. Third, the zone of maturation, or differentiation, where these elongated cells are finally assigned their permanent jobs. They might become xylem, or phloem, or a simple root hair. That's it for the apical meristems. The second type of meristem is the lateral meristems. They are responsible for secondary growth, which is growth in width. This is what makes a woody plant, like a tree, thicker. There are two lateral meristems. Both are cylinders running the length of the stem and root. First is the vascular cambium. It is a thin layer ranging from 1 to several cells in thickness. But it acts as a double-sided factory. When its cells divide, they produce new xylem, or wood, toward the inside, and new phloem, or inner bark, toward the outside. Although this process occurs continuously, during certain seasons growth is more rapid, relatively speaking. In trees, this pattern results in annual growth rings, from which their age and living conditions can be determined, like a very slow wooden diary. The second lateral meristem is the cork cambium, or phellogen. It develops outside of the phloem, and its sole purpose is to produce phellum, which you know as cork. This forms the outer bark, a tough, waterproof, and in fact, dead protective layer for the living tissues inside, similar to your fingernails and toenails if they covered your entire body. There's also a third, less discussed type, the intercalary meristem, found in grasses. These are located at the base of the leaf blades. When you or a grazing animal cut the grass, the apical meristem is removed, but the intercalary meristem remains, and this allows the grass blade to continue growing from the bottom up. Let us admire its quiet, humble persistence. Part 2. The mediators. Hormones. A question I'm never asked is, how do these different meristems know what to do? How is this growth stimulated or inhibited? The answer lies in phytohormones, plant hormones. If you thought hormones only really affected pubescent teenagers and people in menopause or andropause, well, have I got news for you. These chemical messengers are out there wreaking havoc in most higher organisms, including plants. When it comes to plant growth, they mediate what is best described as a slow, decentralized, and deeply bureaucratic process. This area is also insanely complex, so for your listening pleasure, I am only going to grossly oversimplify 5 of the major classes of plant hormones. The most well-known, and perhaps most autocratic, class of plant hormones is the auxins. They are produced primarily in the shoot apical meristem. From there, auxin flows downwards, cell to cell. Auxins have numerous roles, one of which is ensuring apical dominance. Sounds kind of macho, doesn't it? Well, I guess it sort of is. The auxin flowing down from the top shoot actively inhibits the growth of the lateral buds further down the stem. The top bud is chemically shouting, "Hold it right there," to all the potential side branches. This is why a pine tree is shaped like a cone. Another of auxin's primary jobs is to stimulate cell elongation in the shoot. It does this by activating enzymes called expansins, which function as tiny molecular scissors, loosening the cellulose fibers. The cell's internal water pressure, its turgor, then pushes outward, stretching the newly pliable cell wall, making it longer. But auxins do not work alone. They are in a constant, slow-motion political battle with the cytokinins. Cytokinins promote cytokinesis, or cell division. They are produced primarily in the roots and travel upwards. Like the proportion of Democrats to Republicans in the House and Senate, the ratio of auxin to cytokinin determines what happens. A high auxin to cytokinin ratio signals root generation. A low auxin to cytokinin ratio signals shoot induction. This chemical negotiation dictates the entire plant's architecture. Third, we have the family of plant hormones called gibberellins. Besides having a name that is delightful to say, gibberellins are the hormones of drama. They promote dramatic stem elongation. This is the hormone responsible for bolting. No, not when the plant runs away, but for example, when a lettuce plant suddenly shoots a tall flower stalk. Gibberellins are also crucial for breaking seed dormancy. They are the go signal that tells the embryo it is time to grow. Fourth, we must discuss inhibition. Growth cannot be unregulated, and this is the role of abscisic acid, or ABA. ABA is a stress hormone. It is the plant's emergency brake. ABA enforces dormancy in seeds and buds, preventing them from growing until they are ready. When a plant is under drought stress, it is ABA that floods the system, signaling the stomata, or the leaf pores, to slam shut, conserving water. Finally, we have ethylene, which is actually a gas. Ethylene is the hormone of aging and ripening. It is a communal signal. One ripening fruit, a banana perhaps, releases ethylene gas, which signals all the other bananas around it to also ripen. This is responsible for the one bad apple effect. Ethylene is also responsible for abscission, the deliberate shedding of leaves or fruit. It does this by promoting the breakdown of cells in a specific predetermined abscission zone at the base of the leaf stalk. It is a controlled and orderly amputation. Part 3. The navigational system. Tropisms. We have our growth engines, the meristems, and our chemical signals, the hormones. Now, how do these plant parts know which way is up and which way is down, or which way the light is coming from? This is the study of tropisms, growth in response to an external stimulus. Given plants' general lack of sensory organs as we know them, for example, eyes, this is by far one of the coolest things that plants do, in my humble opinion. So of course I left it for last to avoid exciting everyone too much. Let's begin with gravitropism, the response to gravity. The shoot exhibits negative gravitropism. It grows away from the pull of gravity. The root exhibits positive gravitropism. It grows towards the pull of gravity. But how? Well, the mechanism is beautifully simple and entirely passive. Inside specific cells in the root cap and in the stem are dense, starch-filled organelles called amyloplasts, or statoliths. These statoliths are heavy, heavier than the rest of the cell's contents, and so they settle. They sink to whatever part of the cell is down. They are the plant's tiny internal gravity detectors. In the root cap, When the statoliths settle on the bottom side of a cell, they trigger a redistribution of auxin. Auxin is actively pumped to the bottom side of the root tip. Here is the critical detail. In a root, high concentrations of auxin inhibit cell elongation. This is the opposite of what it does in the shoot. So the cells on the top of the root with less auxin elongate normally. The cells on the bottom of the root with more auxin are inhibited and grow more slowly. The result? The top grows faster than the bottom, and the root bends downwards. Now the shoot. A similar but opposite event. If a plant finds itself lying on its side, the statoliths in the stem cells settle. Auxin is also redistributed to the bottom side of the stem. However, in a shoot, auxin stimulates cell elongation. So the cells on the bottom of the stem elongate faster, while the cells on the top elongate more slowly, leading the shoot to bend upwards. The same hormone, the same redistribution, but an opposite differential response. What a very frugal and potentially confusing system. This same principle applies to phototropism, growth towards light. When light, particularly blue light, hits one side of a shoot tip, it causes the auxin on that side to migrate away toward the shaded side. The shaded side now contains a higher concentration of auxin. As we know, in a shoot, auxin stimulates elongation. The shaded side grows faster, while the illuminated side grows more slowly, and the stem inevitably bends toward the light. Finally, we have thigmotropism, a response to touch. This is how a vine's tendrils find and climb a support. When a tendril gently touches an object, the cells on the side not in contact begin to elongate faster. The cells that are touching are inhibited. This differential growth causes the tendril to curl slowly and firmly around the object. A quiet, gradual entangling, as I hope sleep has long since grasped your mind. And, pun very much intended, that wraps up our discussion of plant growth. A bureaucracy, a constant silent negotiation between push and pull, start and stop, up and down. It is a complex, decentralized, and wonderfully predictable set of procedures, all happening at a pace perfectly suited for sleep. Thanks for tuning in to Science Me to Sleep. See you in the next chapter.